I'm back, Rainiel Aquino Sarmiento, third year college, BS Criminology, and this video presentation is all about DNA analysis. Okay, let's start. Objectives. Lesson objectives explain the importance of DNA analysis and enumerate the steps in DNA analysis. We're going to the second slide. Second slide, describe DNA, its location, composition, and nature. Okay, DNA. DNA or deoxyribonucleic acid is self-replicating material which is present in nearly all living organisms as the main of constituent of chromosome. It is a carrier of genetic information, and DNA is made up of molecules called nucleotides. Each nucleotides contain a phosphate group of sugar group and nitrogen base. The four types of nitrogen base are adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA instruction or genetic code. The origin of DNA can be considered as a modified form of RNA since the normal pivot sugar in RNA is reduced into deoxyribose in DNA, whereas the simple base uracil is methylated into thymidine. Now we're going to the third slide. Describe forensic DNA analysis and how it's used to solve crimes. Forensic DNA analysis is the use of deoxyribonucleic acid specimen in legal proceedings, just as people can leave fingerprint when they touch surface, so too can they leave biological material that contain DNA. DNA is generally used to solve crime in one of two ways. First, in cases where a suspect is identified, a sample of the person's DNA can be combined to evidence from the crime scene. The result of the comparison may help establish whether the suspect committed the crime. The second is in cases where a suspect has not yet been identified, biological evidence from the crime scene can be analyzed and compared to offender profile in DNA database to help identify the perpetrator. Crime scene evidence can also uh, be linked to other crime scene through the use of DNA database. Okay, we're going to the fourth slide. The fourth slide is describe the four steps in DNA analysis. Steps in DNA analysis. They have four several basic steps are performed during the DNA testing regardless of the type of test being done. The general procedure includes first, the isolation of DNA. From an evidence sample containing DNA of unknown origin, the general at a intertime the isolation of DNA from a sample from no individual. The second is the processing of the DNA so that the result may be obtained. The third is the determination of the variation in DNA test result from the specific regions of the DNA. And the fourth and the last is the comparison and interpretation of the test result from the unknown and known sample to determine whether the known individual is not the source of the DNA or is included as a possible source of DNA. While you are going the last slide, the last slide is describe the three types of DNA evidence analysis. Okay, types of DNA evidence analysis. The first type is polymerase chain reaction. The copying process known as the polymerase chain reaction uses an enzyme to replicate DNA region in a test tube. Mm, by repeating a copying process, a small number of DNA molecule can be reliably increased up to billion within the several hours. RFL analysis require a biological sample about the size of quantity, but PCR can be used to reproduce millions of couples of the DNA contained in a few skin cells. Since PCR analysis require 1 minute quantity of DNA, it can enable the laboratory to analyze highly degraded evidence for DNA. On the other hand, mm, because the sensitive PCR technique replicates any and all of the DNA contained in a evidence sample greater intention. To contamination issue is necessary when identifying, collecting, and preserving DNA evidence. This factory may be particularly important in evaluation of unsolved cases in which evidence might have been improperly collected or stored. Uh, the second is STR or short tandem repeats. Technology is forensic analysis that evaluates specific regions that are found at the nuclear DNA. The variable nature of the STR region that are analyzed for forensic testing intensify the discrimination between one DNA profile and another. For example, the likelihood that any two individuals except identical twins will have the same 13 lossy DNA profile can be as high as 1 in 1 billion of greater. Okay, the third is Y chromosome. Y chromosome markers target only the male fraction of the biological sample. Therefore, this technique can be very valuable if the laboratory detects complex mixture within a biological evidence sample. Because the Y chromosome is transmitted directly from father to all his son, it can also be used to trace family relationship among uh, among mates. Advancement in Y chromosome testing may be eventually eliminate the need for laboratories to extract the separate semen and vaginal cell. For example, from a vaginal swab of a rape kit prior to analysis. Uh, Mitochondrial DNA. This analysis allow forensic laboratories to develop DNA profile from evidence that may not be suitable for RFLP or STR analysis. FT DNA technology analyze DNA found in a different part of the cell. Okay, is the end of my video presentation. I hope you learned. Thank you for listening. Uh, Thank you, ma'am. God bless.